Hello, everyone. I'm Aditi Doddavaram, and today I'm so excited to be talking to you about the mechanisms of feedback disinhibition in synesthesia and near death experiences. We'll be talking about what synesthesia and near death experiences are and why they can be connected through feedback disinhibition. A little about myself to start with. I live in Bangalore in India, and I've always had a deep interest in neuroscience and cortical processing. And the uniqueness of the experiences created by synesthesia and NDEs is what led me to pursue this research. So, first, let's start off with what synesthesia and near death experiences actually are, because it's quite likely that most people have never heard those words before. Synesthesia involves a connection between two different sensory streams, meaning that stimulation of one sensory stream, called the inducer, leads to the involuntary stimulation of another, which is called the concurrent. This can often lead to strange experiences, such as being able to hear colors or smell sound. Near death experiences, on the other hand, are profound transcendental experiences that occur on the threshold of death. They usually occur during conditions during which the body is under extreme stress, such as cardiac arrest, and can create things like out of body experiences or visions of heaven. While these two perceptions may seem completely disconnected from one another, there is one extremely important way we can connect them: cortical feedback disinhibition. Both synesthesia and NDEs could be caused by feedback disinhibition between different parts of the brain. But what is feedback disinhibition? First, let's talk about feedback inhibition and how a disruption in this balance can lead to disinhibition, which could be normal or abnormal, as in synesthesia and NDEs. Feedback inhibition is important for regulating cortical excitation and processing sensory perception. It occurs through a self limiting pathway where excitatory cells release glutamate, which stimulates the GABAergic neurons, causing them to secrete GABA. This stimulates the GABA sensitive receptors, which then inhibit the original pyramidal cells. However, this type of inhibitory mechanism can be broken if the original inhibitory cells are themselves inhibited, creating a double negative effect. This is what happens in feedback disinhibition. Feedback disinhibition can be driven by different antagonists that act on the inhibitory interneurons. These antagonists could be other interneurons or neuromodulators like acetylcholine. This disinhibition could be normal, as in the case of learning fear responses, since it would be difficult to create new neural connections, such as the ones required for learning habitual fight or flight response. With the relatively inactive state created by inhibitory circuits, the disinhibited feedback creates the neural plasticity required to form these new connections. So, how is feedback disinhibition related to synesthesia and NDEs? Well, in many studies, synesthetic individuals have been shown to have modality specific disinhibition between different parts of their brain, leading to cross activation of stimuli and creating these structured sensations. These circuits, such as the ones connecting higher associative areas, such as the visual word processing area, and lower sensory areas, such as the visual cortex, could be responsible for synesthetic experiences, such as the involuntary connection of the letter A to the color red. This type of disinhibition is usually functional, predictable, and permanent. NEAs, on the other hand, have been seen to have global disinhibitory effects, with disinhibition seen between the anterior and posterior areas of the brain caused by acute physiological stress, such as hypoxia. They are largely based on the widespread failure of disinhibitory mechanisms due to energy failure, creating uncontrolled global disinhibition. This disinhibition creates large scale sensory overload, leading to the dramatic hallucinatory experiences associated with NDEs. While these theories may or may not be true, it's important to make sure that they are testable. We can do this through two methods. One, which has often been used, is to use drugs known to have disinhibitory effects to create synesthesia adjacent experiences or near death adjacent experiences. These drugs for synesthesia include LSDs, which create an approximation of synesthetic experiences. The drugs used to simulate NDEs include ketamine, which closely approximates the effects that NDEs have on the brain. An observation of this has been increased top-down disinhibition in the case of LSD, similar to synesthesia, and anterior-posterior disinhibition in the case of NDEs. If these disinhibitory drugs can reliably induce synesthetic or near-death adjacent experiences, it is quite likely that these disinhibitory mechanisms are the basis for these phenomena. The other method acts as somewhat of an inverse of this method by treating volunteers experiencing these phenomena actively with inhibitory drugs such as benzodiazepines. These can negate the disinhibitory effects that we think synesthesia and NDEs have on the brain and therefore create breaks in these experiences, proving that they are based on disinhibitory mechanisms. So these are a few results from experiments done to observe disinhibited feedback in synesthesia and NDEs. In the first image, we see a specific interconnection between LSA, which is a grapheme processing area, and V4, a color processing region. Here, modulatory connection refers to the influence that LSA has on V4. Here, the connection being measured also shows the activation of V4, which is usually only activated when a color is being perceived. As seen in the graph, this modulatory connection has a positive peak, showing that there is some powerful connection created between these two regions. In synesthetes, disinhibitory mechanisms between these two regions create these connections, leading to structured associations. This creates the perceptual blending associated with grapheme color synesthesia. In the second image, as you can see, there is an increased presence of theta and gamma waves in NDs as compared to the waking state, observed during experiments done on rats. This suggests the presence of disinhibited brain activity during these states, since the theta and gamma wave occurrences are higher in NDs than in the waking state. What's also interesting here is that the theta and gamma waves are responsible for memory encoding and retrieval, as well as attention and perceptual binding, which could be the reason behind this rich episodic experiences created by NDEs. Why is this research important? At first glance, it may seem as though studying two rare phenomena like these is a futile endeavor. But learning about their disinhibitory mechanisms and how they can be induced or treated can help with treating mental health disorders or hallucinogenic states, which are theorized to be based on similar disinhibitory mechanisms. These disorders could include schizophrenia, Parkinson's disease, and PTSD. We can also ground these states in standard biophysical terms, understanding them through brain activity changes rather than only through the first-hand accounts that we currently use to study them. Thank you for listening, everyone. Does anyone have any questions?
Thank you very much for your very informative presentation. Yes, there are some questions coming in. The first is, how did you get interested in this topic?
Like I said, I read about synesthesia a while
Mhm.
ago, and it was a very rare occurrence which kind of interested me. And it's the fact that these are such, interesting rare things that the brain can do, and like it's difficult to observe them, but it's so interesting to do so.
Okay.
So yeah.
What advice would you give to another student who wanted to do similar research?
What advice would I give them? Maybe be very careful when you're going through the research papers that are there, especially on NDEs, because I feel like a lot of them are not, as I said, grounded in standard biophysical terms. Like they're mostly, this person said this, or this might be what is behind this, but the proof we have is that this person thought this. So what you should do is look for papers which say, in NDEs in terms of this brain mechanism, NDEs in biophysical terms, stuff like that. So you can ground your research in standard terms. Yeah.
Great. Well, thank you very much for your presentation. I feel like I learned a lot.